Hi guys, in this lesson we're going to look a little bit more closely at the cell cycle and the steps of the cell cycle. So we're going to start with kind of an introduction overview of those steps and then go a little bit more in depth into each of those steps as we go. So the life cycle of a cell is divided into two major portions. Okay. So those two major portions are first interphase and the second is cell division. Now, cell division includes a couple different parts. So the first part of cell division is mitosis. So mitosis is where we make copies of DNA and divide up that DNA so then each new cell that we make has the same genetic information. Okay, so that's mitosis. Then the second part of that is cytokinesis. So this is the division of the cytoplasm and everything else inside of the cell. And then at the end of cytokinesis, we officially have two brand new cells. Okay. Now, important to remember, the process of cell division is continuous. So it's not like a stop and start each step. Okay, it's a fluid process. We only divide it into these stages for like our convenience to understand what cells do and to help you guys learn this a little bit better and understand this a little bit better. Okay. So it's not that it does one step and then it stops and then moves to the next step and stops. Okay, it continuously moves through all of those steps in kind of like a fluid motion. Okay, but we just break it down in those steps so it's easier to understand. So this is a diagram that we will usually use to talk about cell division. Notice the different sizes of each of these steps. So those sizes show you how long each of those steps take. So you can see from this diagram that most of the cell cycle happens with interphase. So interphase is the part of the cell cycle that takes the longest. This is kind of what we call the like growth and maintenance part of a cell's life cycle. Okay, interphase is where a cell is hanging out most of the time. Then it takes a little decent chunk to do mitosis, and then cytokinesis is sometimes said to be a part of mitosis. They kind of happen at the same time, but it's just kind of like a little blip on the radar here. It's just a tiny little quick process that officially divides us into two new cells. Okay. Notice over here this G0 phase, okay? This is when the cell cycle stops, so that's kind of a special case. So we're not necessarily going to be talking about that phase as much. We're going to be focusing on the ones that are in this circle here in this lesson. So we're going to start with interphase, the longest phase of the cell cycle. So in interphase, we have cell growth and DNA replication. So the cell is growing and maturing, and the DNA has copies of it being made. Okay. This is not considered a part of mitosis. That is very important to remember. Okay. Interphase is a phase completely separate from the stages of mitosis. So interphase actually represents a majority of a cell's life cycle, like we said, as we saw in that diagram on the previous slide. Okay? And interphase includes a couple different phases within it. Okay, so we're going to talk about a lot of different phases here. One of the main parts that's included in these phases is cell growth and maintenance, so making sure that things are working properly in the cell and fixing them if they're not. The duplication of DNA. Okay, and in this stage, DNA appears in a form that we call chromatin. So chromatin is this kind of not super tightly wound DNA. It hasn't become chromosomes yet. That'll make a little bit more sense when we talk about that in the stages of mitosis. Okay? And the last thing that it includes, I guess we could say, is that the organelles are going to replicate in interphase as well. So basically, interphase is the preparation for mitosis and for actual cell division. Interphase is all of the steps that you need to do in order for mitosis to happen properly. You can kind of think of it as like if mitosis were like our official moving day, moving into a new house or something like that. Interphase is all of the prep, getting ready to move. So you're packing up all the stuff that you need to pack up, making sure that it's packed nice and safely, and getting it all into boxes, labeling those boxes so you know where they're going to go and that kind of stuff. Okay. So we can kind of consider interphase that preparation phase. Okay. So the three phases that we divide interphase into are the G1, S, and G2 phase. Okay. So we're going to go through each of these phases of interphase in a little bit more detail here so we can have an idea of what we're accomplishing in each of these steps in interphase. So we're going to start with the G1 phase. Now G1 stands for first growth phase. So we have two growth phases and then a synthesis phase. So the G1 phase occurs after a cell has undergone cell division. It's the very first thing that a cell starts doing as soon as it has divided into a new cell. So cells begin to mature. They increase in size a little bit by making more cytoplasm, because when they first divide, they're a little bit small. They have room to grow, but remember, they can't grow super big because of those limits to cell growth. Okay, their volume can't get too big because the surface area will be way too big. Okay, and also we don't want to have too much space that we can't make enough proteins and nutrients and stuff for it. Okay, so the cell will increase in size by making more cytoplasm. It can also synthesize new organelles, and also it's just going to carry out all of the normal metabolic activities that they need to. So they're going to be taking in their food and making the nutrients that they need in this process. Next, after the G1 phase is the S phase, which stands for synthesis phase. Now, this is the phase where the DNA is copied or replicated. You will usually hear it described that way too. So it's important to remember that DNA is copied before mitosis even happens. Okay, so once we have those copies of the DNA made, then mitosis is responsible for making sure that each of the new cells gets the correct DNA in them. Then the last phase of interphase is the G2, which is the second growth phase. So this is where the cell makes all of the structures that it needs to divide. So that would be any organelles that need copies made of it. This is where centrioles are going to come into play in animal cells for cell division. They're going to help organize and get everything ready and tell everything where to go when it comes to mitosis starting. Okay. So now we have everything in order, getting ready to hop into mitosis, the actual kind of real cell division, I guess you could say. So this is what that kind of looks like for each of those phases. For interphase, we see that like you can't really tell that there's any chromosomes or anything. This stringy kind of purpley stuff in the nucleus of all of these cells. So this little circle here is the nucleus of both of these cells. Notice that is the DNA there that we can see, that purple stringy kind of stuff. It doesn't have any defined shape. Okay, that's how you know that it's an interphase. Okay, we don't have any super defined shape to the DNA at this point. Now, when we look at pictures of cells in later stages, you are definitely going to see a difference in the shape of that DNA later on. So remember this picture when we get to looking at those other ones, because it is going to be a big difference. Here's kind of the cartoon version of it. Usually these cartoon ones are a little bit easier to see what's going on. Okay, so there's no super defined DNA in this nucleus. All of this like fuzzy stuff around here, this is the chromatin. So this is the DNA just kind of floating around in the nucleus. Copies of the DNA are probably being made here. And And then notice that the centrioles are starting to appear. Okay, so this is an animal cell. Centrioles are only found in animal cells. And centrioles are kind of like the conductors of cell division in animal cells. They tell everyone where to go. You can think of it like a stage director or something like that, or stage manager. They tell all the actors where to go, when to go, that kind of stuff. Centrioles tell organelles where to go and when to go, and tell the nucleus to divide, tell the cytoplasm to divide, all of that good stuff. Now, when we are talking about cell division with the cell cycle, we divide that cell division into two parts, like we said earlier, mitosis and cytokinesis. Okay, so we're going to talk about these in a separate lesson, but to give you a little bit of an introduction into that, mitosis is the division of the nucleus in the genetic material. Okay, so this is strictly the division of DNA and making sure that each new cell that we make has the appropriate amount of DNA. Because if it doesn't have the correct DNA, doesn't have the right amount, then we're going to have some problems. Okay? So mitosis is strictly the division of the genetic information. Then, after mitosis has completed, cytokinesis can happen. So this is when the cytoplasm divides. So this is when we officially have two new daughter cells formed. Okay. So the new membranes form, they close off on each other, and then you have two brand new cells sitting there ready to do what they need to do. And then once those cells have divided, the process starts all over again. So they hop right into that G1 phase, move on to the S, G2, back to mitosis, divide into more cells, and it just keeps on going until they're told to stop, or until they reach a point that they can't do it anymore. If they get damaged, they might not be able to divide or something like that. So we'll talk about some of those things that may cause that to happen later on in our later lessons. But that is going to do it for this one. Our next lesson, we're going to cover mitosis in depth and look at all of the steps of mitosis. So get ready for that. I'm looking forward to it. And make sure that your electronic copy of your notes for this gets filled in.